Hello, everyone. Welcome to Global IMMUNOTALK 2024. I'm Kazuyo Moro from Osaka University and Riken, Japan. Today, I'm so excited to welcome Dr. Shizuo Akira for his talk. But before moving to his talk, I would like to have two housekeeping announcements. One is about next week's speaker. We will be welcoming Peter Bluding from Karolinska Institute, who has done remarkable work on human immunology. So please join us again. And the another announcement is about how to make a question in this seminar series. Unfortunately, we cannot directly get the question. So if you have a question to the speaker, please use the Twitter, which I'm gonna explain later. Okay, so now I would like to introduce today's speaker, Dr. Shizuo Akira. Shizuo is a director of the Center for Advanced Modality and Drug Delivery System, as well as a special appointed professor at Immunology Frontier Research Center, Osaka University. So he earned his MD and PhD from Osaka University. And after 2 years of the postdoctoral research in the Department of Immunology at the University of California, Berkeley, he began studying IL-16 regulation and signaling at Osaka University. And he cloned the transcription factor NF-IL6, which is sometimes called CEBP beta. And also he cloned STAT3. And from 1996 to 1999, he served as a professor at Hyogo University, where he began his research on toll-like receptors. So by generating so many TLR family knockout mice, he identified ligands for many TLR members. And he also demonstrated that differences in signaling pathway among TLR are due to the selective use of adapter molecules such as MID88 or TRIF. And there are countless paper about TLR, but I personally believe that his biggest contribution is in this field is that he generates so many knockout mice by himself, and but he never hesitates to share these mice to the researcher in other country and also in Japan. So this explains his wonderful character, and I really respect this point. And his research interested later shifted to the effector phase of toll-like receptor responses, leading to the study on the role of messenger RNA degradation in the inflammation and immune responses. So he's currently focusing on the role of the Regnase-1 in a variety of biological process. So now I'd like to call Shizuo. Shizuo, could you please turn on your mic and camera? I think it just, okay. Oh, you disappeared. One click is, I think, enough. And wait for a while. Yes, perfect. Thank you very much. Okay, so now thank you very much for joining to this seminar series. Yes. And
thank you for your kind introduction and inviting me to this global talk.
Thank you very much. And before we listen to your talk, we used to make one question to know about your character. And today I would like to ask you about what kind of what is the most impactful decision which you made in the past in your scientific career is my question. Could you please answer to this question?
The most impactful decision I made was when I became a professor at Hyogo College of Medicine. I have applied for several university professor positions since I have completed my research at Kishimoto Laboratory. However, I was not accepted. Finally, I became a professor of a private medical college. At that time, I felt the desire to abandon all the research I had been doing in the previous laboratory and to develop a new field of my own. So I used knockout mouse as a tool of searching for a theme, and eventually I was able to enter the field of innate immunity. If I had been selected for the professorship I initially wanted, this decision would not have happened, and I would have continued my formal research. From the beginning, my research, I have always been thinking about having my own territory in the research. Yeah.
Wow, that's a big story. Yeah, yeah. I think many young people who is looking for the professor position was really encouraged by your story. And I believe so many university are regulating about their decision in the past.
Be always optimistic.
Yeah, right. Okay, so now let's move to your talk. Could you please share the slide? Please take a time. Okay, and now you need to change to the slide mode. Perfect. Okay, so I'm gonna disappear. So could you, yeah, please start your presentation.
Today I'll talk about the endoribonuclease Regnase-1, a key regulator of innate immunity and metabolism. As you know, toll-like family members recognize different microbial components as shown in this slide. Relatively small number of family members sense both bacteria and viral invasion into the body. Similarly, the signaling pathway also different each other. The difference in signaling pathway among total family members is due to selective usage of adapter molecule, mediated essential all TR signaling pathway except TR3, mediated activate NF-kappa B and lead to induction inflammatory cytokine. So mediated dependent pathway is associated with inflammatory responses. In addition to mediated dependent pathway, there is mediated independent pathway TR3 and TR signaling pathway. This pathway is mediated by another adapter molecule named TRIF. TRIF activated non-canonical IKK, TBK1, IKKI, then this kinase phosphorate IRF3, IRF3 form a dimer that translocate from the cytoplasm to the nuclei and induction of type I interferon. So TRIF-dependent pathway is involved in antiviral responses. So TR ligand identified and TR signaling pathway has been clarified. So we move to the effector phase. That is, what is the role of the genes induced in response to TR ligand? Particularly focusing on the molecule named GC3H12A. These genes induced in in macrophage in response to LPS in rapidly. And this induction entirely depended on MyD88, not TRIP-dependent pathway. So this is 3H12A has a CCH type zinc finger in the middle and form a family with this 3H12B, 12C, and 12D. CCH zinc finger protein function as RNA binding proteins. CCH zinc finger proteins are possibly involved in RNA modification, such as mRNA splicing and mRNA decay. For instance, tetracycline, TTP, are member of C3H zinc finger protein family. They stabilize TNF mRNA via A-rich element in the 3' UTR. So to know the role of Regnase-1, we generate G3A, we generate G3A knockout mouse. This mouse born alive. However, all mouse died within 12 weeks after birth. This mouse showed severe splenomegaly and lymphadenopathy. And plasma cells accumulate in the lung and spleen and lymph node. And also the serum level of immunoglobulin increased, and all isotype is increased in this level knockout mouse. And also, autoantibody is also detected, ANA and anti-double-strand DNA antibody detected in G3H12A knockout mouse. So we first examine the cytokine production, peritoneal macrophage stimulated TL ligand, and examine the production of IL-6 and IL-12P40. I'll show in this slide production of IL-6 and IL-12P40 is significantly upregulated in G6 deficient macrophage in response to all TL ligand. So G612A family members expected to involved in mRNA stability. So we prepared the peritoneal macrophage and stimulated for 2 hours with LPS, then followed by actimation treatment to stop transcription. And then this subject to Northern blot analysis. I'll show in this slide, IL-6 half-life is prolonged. However, in TNF, chemokine KC, is half-life not changed between wild type and this knockout mouse, the deficient macrophages. So this indicates that IL-6 is destabilized by G12A. So usually the MR stability is regulated by 3' UTR. IL-6 3' UTR contains 1 stem loop structure and 5 AU-rich elements. So we constructed a variety of luciferase reporter plasmid constructs in which various portions of IL-3' were inserted in downstream of luciferase genes, and this construct transfecting the cell without G12 expression vectors. In the case of this construct, addition of the expression of G12 expression vector did not change the luciferase activity between without this, there are no big difference in luciferase activity. However, in the case of this construct contains stem loop structure. This construct addition of G12 expression vectors suppress the luciferase activities. So, over this stem loop structure responsible for destabilization by H12A. In fact, only this stem structure is enough to suppress the mRNA by G12A. So this is some other previous result. G12A is an inducible gene response to TAR stimulation. This H12A has a CCH type zinc finger motif and form a family 12B, 12C, and D. These 12A degraded several inflammatory associated mRNAs, including ILC mRNA and its own mRNA via its 3' nuclear stem loop structure. These 12A harbors an N-terminal nucleus domain. These 12A knockout must show that generalized chronic inflammation. Based on the function, we renamed this molecule Regnase-1. However, originally identified the inducible genes, the already Regnase protein present in unstimulated condition. After LPS stimulation, band is shifted and disappeared and reappeared at 4-hour time point after stimulation. Similarly, the IL beta stimulation shows a similar pattern. This pattern is very similar to the ICAP-B alpha degradation. However, TNF does not modify Regnase-1. This molecule also degrade ICAP-B alpha. So we identify the C-terminal portion, IKT consensus motif. DSGXS motif present in C-terminal of Regnase-1. In fact, that this motif is in vitro experiment show that IKK phosphorate this site. And so this summarized previous result in the unstimulated condition, only Regnase-1 is present and constantly degrade IL-6 mRNA and also its own mRNA. After TAR stimulation, MyD activated and IRAK1 activated, then due to IKK complex is activated. Then IKK phosphorate to Regnase-1 and followed by evacuation and lead to degradation. At the same time, ICAP alpha is also phosphorated and lead to the degradation of this molecule. And then Regnase-1 function is shut off. Then NF-kappa-B migrated to the nucleus, then induce induction of IL-6. At the same time, Regnase-1 is also mRNA also induced and protein is produced and degrade both ILC mRNA and Regnase-1 mRNA. So in the Regnase-1 is act as a break in unstimulated cell, and after TR stimulation act as a negative feedback. So what is the difference between TNF? What is the reason that why TNF does not modify the Regnase-1? This slide shows the comparison between IL-1 TLR and TNF signaling pathways. The big difference among these signaling pathways is the presence of IRAK family members in IL-1 and IL-4 signaling pathway. In fact, IRAK is phosphorate Regnase-1. As shown in this slide, in the retarded band appeared. This band is a complete evolutionary phosphatase treatment. And Regnase-1 expression vector, Regnase-1 and IKK beta co-expression induce the faint ubiquitination of Regnase-1. However, in this condition, IRAK1 expression of IRAK1 dramatically induce ubiquitination of Regnase-1. This indicates IRAK1 facilitate IKK beta mediated ubiquitination of Regnase-1. We identified the initial phosphorylation site between IKK phosphorylation site and C-terminal region. This region is initial phosphorylation site. And after TLR and IL-1, IL-17 simulations, Regnase-1 is phosphorated by MyD88 to dependently activated IRAKs or ACT1 dependent activated TBK-IKKI. Then Regnase-1 detached from the ribosome, and this detachment shut off the Regnase function. Then followed by phosphorylation by IKK and lead to degradation, ubiquitin dependent degradations. So IL-17 stimulation induced band shift. However, no disappearance of band as observed in LPS simulations. However, we generate knock-in mouse in in the IKK phosphorylated site mutated to the alanine. In this case, with the after IL-17 stimulation, the phosphorylated band amount of phosphorylated band is increased. This indicates that Regnase undergoes a constant cycle of degradation and synthesis during IL-17 stimulation. So IL-17 stimulation alone does not induce mRNA. When MEF is treated TNF for 2 hours, then wash out, followed by IL-17 stimulation. In this case, the IL-mRNA dramatically increased, accompanied by Regnase-1 phosphorylation. This indicates that IL-6 induction by IL-17 mainly depend on mRNA stability. So the synergistic action between IL-17 and TNF has been clarified. TNF-induced TNF-dependent activation of the promoter of I-kappa beta gene. However, I-kappa beta mRNA constantly degrades Regnase-1. However, IL-17 stimulation shut off the Regnase-1 function and I-kappa beta protein produced. Then this protein binds to P50 and NF-kappa B, and these activate promoter of IL-6, IL-8, lipokine 2, and these messenger are induced. At the same time, this messenger also regulated the Regnase-1. So cytokine network drives the keratinocyte hyperproliferation and skin inflammation characteristic of psoriasis. Iron beta, IL-6, IL-12, GCS, and certain chemokines mRNA, as well as IL-1, IL-17, IL-18, IL-33, and IL-36 signaling pathway are regulated by Regnase-1. Many papers reported the role of Regnase-1 in pathogenesis of psoriasis. And group 2 innate lymphocyte cell ILC2 is a mediator of allergic inflammation. Upon allergens or infection, lung epithelial cells produce IL-33 and IL-25, TSLP, and IL-33 and IL-25 act on IL-3 and produce IL-13 and IL-5, IL-13 involved in goblet cell hyperplasia and mucus production. On the other hand, IL-5 is involved in erogenous trafficking. And IL-33 is an IL-1 family cytokine which activates mediator-dependent pathway, and IL-25 is an IL-17 family cytokine which activates ACT1 TBK-IKK1 signaling pathway. So we speculated that Regnase-1 is also involved in ILC2 activation. So control mouse and Regnase-A mutant mouse, which enhance the Regnase-1 activity and inoculated with nasally inoculated with IL-33 for 3 days. Then analyze at 4 days. We analyze this the lung, and ILC2 number is significantly decreased in Regnase-A mutant mouse, and IL-5 and IL-13 production in BAP is also significantly suppressed, and mucus protein production also is suppressed. A similar finding also observed in when we treated the mouse with IL-25. So this shows that Regnase-1 is regulate the ILC mediated type 2 immune responses. So we generate a T-cell specific Regnase-1 knockout mouse. This mouse showed born alive, and however, all mouse died within 17 weeks after birth. And this mouse also showed severe splenomegaly and lymphadenopathy. And this splenic T cell, CD4 T cell stimulated PMA plus ionomycin, as shown in this slide, interferon gamma producing CD4 T cell and IL-17 producing CD4 T cell, IL4 producing CD4 T cell, all increase in the number. More interesting is usually in the CD4 T cell, major population is the naive type. However, in the case of Regnase deficient CD4 T cell, most effector and memory types. And also the CD69 activation tissue retention marker is also upregulated. And plasma cell also increased in spleen of the knockout mouse. So we transfer the CD25, 5.2 plus Regnase-1 deficient CD4 T cell into CD45 1 point recipient mouse. And after 3 months later, we performed the FACS analysis. In the case of transfer of the Regnase-1 deficient CD4 T cell, this T cell is proliferated and survived. However, control normal T cell do not proliferate and disappear. And interestingly, in this recipient mouse shows the change of the naive T cell into effector memory CD4 T cell of recipient origin, and plasma cell also increased. This CD44 Regnase deficient CD45.1 positive recipient mouse induce the same phenotype in the recipient. In the CD5 2-point Regnase-1 deficient CD4 cell induce the same phenotype in the recipient mouse as the T cell specific knockout mouse. So recently, Regnase-1 found as a negative regulator of T cell function within the tumor environment. And Regnase-1 null CD8 T cell persist longer in tumors and express more effector molecule. And Regnase deficient T cell upregulate expression, BATF and TCF. This induces enhanced killing activity and cell longevity. So Regnase deficient is deficient CAR T cell is maybe useful for cancer immunotherapy. Different from macrophage, and T-cell stimulation did not induce phosphorylation or disappearance of the band. As the amount of Regnase-1 protein decreased, a new fast migrating band appeared. And this slide shows a comparison between TL T-cell syndromic pathway. In T-cell syndromic pathway, T-cell activation induce CBM complex via PKC-theta. CBM complex composed KALMA1 BCL10 MOLT1. And MOLT1 is reported as a protease, so we speculated MOLT1 is involved in Regnase cleavage. In fact, overexpression MOLT1 cleaves the Regnase-1. So in unresting condition, the Regnase-1 constantly regulate its target mRNA. However, T-cell stimulation, MOLT1 activated, MOLT1 cut Regnase-1 and shut off the Regnase function, and IL-2 mRNA induced and induced the proliferation of T cells. We recently generated NK-specific Regnase-1 knockout mouse, and we first examined the interferon gamma and granzyme production with cytokine simulation, as shown in this slide. And the interferon gamma production and granzyme production is enhanced in Regnase-1 deficient NK cells. And we next examine the in vitro killing activity when co-cultured with B16F10 melanoma cells. Regnase-1 deficient NK cell showed more killing activity against B16 B16F10 cells than control NK cells. And during the culture, interferon gamma production is increased in Regnase deficient NK cells. So we next examine the in vivo anti-tumor activities, B16F10 subcutaneous injected and tumor size was measured. As shown in this slide, Regnase-1 delta NK mouse strongly suppress the growth of B16F10 melanoma cells. And also we perform the metastatic effect on the metastasis of tumor cells. As shown in this slide, Regnase delta NK mouse show the suppress the metastasis to the lung of B16F10, as shown in this slide. And next we performed the cytoanalysis of the immune cells inside the tumor. As shown in this slide, NK cell population is increased in tumor of the Regnase delta NK mouse, and also in the CD8 T cell and the CD4 T cell number is also increased. And this immunoblotting show that in the case of the Regnase delta NK, tumor has necrotic lesions, and NK cell more NK cell accumulated in Regnase delta NK mouse, and also this NK cell highly expressed powerfully. Then we prepared these tumors and isolated the RNA to examine the expression of cytotoxic associated genes, as shown in this slide: granzyme A, granzyme B, granzyme K, and interferon gamma. Powerfully is upregulated in Regnase deficient the tumor from the Reg1 delta NK mouse. On the other hand, the angiogenesis associated VEG alpha F alpha is also suppressed. We prepared from the NK cells and performs we prepare from the BC2F tumor tissue from the control and delta NK cell and perform single cell RNA-seq analysis. In the case of delta NK cell, some cluster of the tumor is disappeared, and NK is number is increased. And in this NK from the delta NK cell highly expressed the tumor-cidal gene, granzyme, and papain. Next, we prepared from the PIL-NK from the tumor-bearing mouse. These PIL-NK show the completely different pattern of the clusters between control and delta NK mouse. Particularly focused on cluster 6. Cluster 6 is highly expressed tumor-cidal associated genes. In addition, this cluster express CXCR6. So we next compare the chemokine receptor between tumor-infiltrating NK between wild type and reg delta NK mouse. CXCR6 is specifically expressed in PIL-NK of Regnase-1 delta NK mouse. And we next perform the FACS analysis in the spleen NK cells. Most abundant NK cell express CXCR6 in Regnase-1 delta NK mouse. More interestingly, the PIL delta NK, most this NK cell express CXCR6. So we next CR6 is the actual target of Regnase-1. In fact, this Lucifer's activity shows the Regnase-1, CXCR6 is the direct target of Regnase-1. So the ligand of CXCR6 is CXCR16. This CXCR16 is highly expressed in tumor from delta NK mice. And then this CXCR6 ligand CXCR is highly expressed in macrophages and dishes inside the tumor. So we next to confirm the role of the NK cell in anti-tumor activity, we perform the depletion experiment. Anti-CD3 antibody to deplete the CD8. This does not affect the anti-tumor activity. However, anti-NK1 antibody that depletion of NK cell abolishes the tumor-cidal activity. And similarly, anti-interferon gamma antibody treatment also abolishes the anti-tumor activities. So this clearly shows that the anti-tumor activity observed in reg delta NK mouse is mediated by Regnase efficient NK cell, and particularly interferon gamma produced from NK cell play a critical role for anti-tumor activities. So we next after the interferon blockade, we prepare the tumors and examine the tumor-cidal expression of tumor-cidal genes or the other chemokine receptor and chemokine. As shown, this are interferon gamma, granzyme B, perforin, and CXCR6 and CXCR6. All these messenger completely disappeared. This indicates that interferon gamma play a critical role for infiltration of Regnase deficient NK cell into the tumor. So this is some of the previous results. Regnase deficient NK cell express CXCR6, and this after stimulation, this NK cell produce high amount of interferon gamma, perforin, and granzyme. Then this kills the tumor, and at the same time, interferon gamma act on the tumor microenvironment. These cells produce its ligand CXCR16. Then this mechanism is positive feedback mechanism enhance the anti-tumor activity of Regnase deficient NK cells. So next we try to identify the molecular mechanism of interferon gamma overproduction. We found OCT2 is upregulated in Regnase deficient NK cell in steady state compared to control NK cells. And this OCT1 production is upregulated after cytokine stimulations. And this OCT2 is bound to the ChIP-seq analysis identify this OCT2 binding to interferon promoter region. And I do not show that data, but OCT1 is a real target of Regnase-1. So we previously that ICAP-zeta is also involved in interferon gamma production from NK cells. ICAP-zeta deficient NK cell produce less interferon gamma in response to IL-12, IL-18. And we previously also showed that ICAP-zeta is required for the IL-6 productions. ICAP-zeta bind to NF-kappa-B, and this combination association is activate IL-6 promoter. So this result suggests that ICAP-zeta and NF-kappa-B And also, OCT2 is responsible for interferon production from Regnase deficient NK cells. In fact, in the OCT1 ICAP data, NK form a complex each other, and this OCT2 expression of OCT2 and ICAP data, NK exon vectors synergistically activate IL6 promote interferon gamma promoter activities. And OCT2 binding site is adjacent to the NF-kappa B in the interferon gamma promoter regions. And protein DNA immune complex detects the binding of the protein complex. In the unstimulated conditions, OCT2 is more amount of the OCT2 bind to the interferon gamma promoters. And after cytokine stimulation, the binding more enhanced. And also this similar pattern also observed ICAP zeta proteins. However, NF-kappa B compared P50 and P65 is comparative between wild and Regnase deficient NK cells. And OCT1 deletion of the promoter is completely abolished, OCT1 dependent enhancement of interferon promoter activities. So this slide some other previous result in the interferon promoter after stimulations, OCT2 and ICAP zeta, NK-kappa sit on the interferon beta promoter, and then this induce interferon gamma, produce interferon gamma. So we generate Regnase deficient intestinal epithelial cell knockout mouse. The mouse treated with 2% DDS for 7 days and replaced by waters. As shown in this slide, in the Regnase deficient IEC shows better recovery in this DS model, DS colitis model. On the other hand, Regnase mutant, which enhance the Regnase activity in the aggravated DSS, recovery is impaired. So this slide shows we observe the decrease in panel positive cells and Ki68 positive cell increase. This indicates the Regnase deficiency suppress colon epithelial cell death and promote proliferation during DS treatment. And dapamycin is an inhibitor of mTOR signaling pathways. When we treated the rapamycin, this RNA DDS model, we found that rapamycin treatment aggravated DS-induced colitis, as shown in this slide. And the same weight loss curve, of normal and Regnase deficient mice indicate that enhanced regeneration of colon epithelial cells mainly due to enhanced mTOR activities. In fact, mTOR phosphorylation, mTOR protein level has increased in Regnase deficient intestinal epithelial cells. And also downstream S6K and 4EBP1 and phosphorylation is upregulating the Regnase deficient epithelial cells. So we found that the Regnase-1 regulate mRNA mTOR component genes BR3 prime UTR, as shown this. This Lucas clearly shows the several components of the mTOR component gene is a target of Regnase-1. And ATP is actively released in the extra environment in response to tissue damage and cellular stress. Extracellular ATP enhances tissue repair and promotes the recruitment of immune cells and acts as a co-activator of NLRP3 inflammasome. The conversion of extracellular ATP to adenosine through the enzymatic activity of ectonucleotidases CD39 and CD73 act as a negative feedback mechanism to protect tissue from injury. we found that the expression of CD39 and CD73 and ADORA1 PI receptor is upregulating the Regnase-1 deficient intestinal epithelium. And in addition, I found that metabolism also enhanced in Regnase-1 deficient intestinal epithelial cells. So Regnase-1 regulate mTOR pathways and extraprenage signaling pathways, and also metabolism. So these three knockout mouse enhance this activity. So this Regnase deficient epithelial cell specific Regnase deficient mouse is resistant to DDS induced colitis. So recently, several groups identify mutation IDP patient. They identify the mutation in inflamed mucosa of IBD patient. The genes infected genes are mainly involved in IL-17, PLR signaling pathway, IL-17 receptor, ACT1, and Regnase-1, I-kappa beta, and also PIGR, polymeric immunoglobulin receptor, is highly mutated in inflamed mucosa of IBD patient. So interestingly, the mutation, Regnase-1 mutation occurred in this DSG-IGS, this IKK phosphorylation site. This finding is consistent with our Regnase-A mutant is highly susceptible to DDS-induced colitis. So in the Regnase-1 suppress inflammation by decaying inflammation-associated mRNA, including IL-6, Regnase-1 knockout mouse show a severe autoimmune disease-like manifestation. And in the DSS-induced colitis mouse model, intestinal epithelial specific region of Regnase-1 enhanced tissue degeneration and suppressed inflammation. Regnase-1 acts as a suppressive factor of inflammation in immune cells, whereas it functions as a regulator of mTOR activity and nucleic acid metabolism. Regnase-1 is evolutionarily highly conserved. Regnase-1 exerts different functions in different organisms. In the case of C. elegans, Regnase-1 involved in lipid storage, lacking in C. elegans lacking Regnase-1, worm does not store the lipid. In the case of Drosophila, lacking the Regnase-1 developed to the pupa. However, this mouse did not mature to adult. Regnase-1 is involved in pupa to adult transition. So, Regnase plays an important role in metabolism in addition to inflammation. Muscle D, metabolic dysfunction associated steatotic liver disease is a common chronic disorder highly associated with changing lifestyle of people. The more advanced form of muscle D, known as MASH, characterized by changing immune response and metabolism that lead to progressive fibrosis. The outcome such chronic insult to the liver may conclude with cirrhosis and end-stage liver. Regnase-1 deficient, Regnase-1 is subject to MASH diet 4 weeks, then using AAV clay, deleted the Regnase-1, then subject to further 4 weeks NASH diet, then examine the histology of the liver. As shown in this graph, Regnase deficient liver, the fibrotic decrease in lipid droplet, also decrease in fibrotic change, as shown in this slide. So digital Regnase is prevent the progression of MASH. So this slide summarizes today my talk. Regnase initially identified LPS in dish gene that the knockout mouse show a severe general inflammation. Regnase is an endoribonuclease and regulate MRS-associated inflammation. Regnase protein is present in unstimulated immune cells and become regulated in mediated-dependent manner in response to TR ligand, but not TNF. The DSGXS motif is present in the C-terminal region of Regnase-1 and phosphorylated by IKK and regulated in ubiquitin proteasome-dependent manner. Regnase co-localizes with ribosome and suppresses translation active mRNAs. Phosphorylation of IKK consensus is necessary for degradation of Regnase, but dispensable for the shut of Regnase-1 function. IL-17 stimulation phosphorylate Regnase-1 here up to one PBK-IKK axis, which induce release of Regnase-1 from the ER. T-cell specific Regnase-1 knockout mouse showed severe inflammation characterized by accumulation of effector memory T cells and plasma cells. Regnase-1 is cleaved by MALT1 in TCR-stimulated T cells. Regnase-1 play an important role ILC-mediated allergy. Regnase-1 deficient NK cells show strong anti-tumor response by high production of anti-tumor molecules and effective recruitment of NK cells. Colon epithelial-specific division of Regnase-1 shows the resistant DS-induced growth, deactivation of mTOR and pre-metabolism. Liver-specific division shows the resistance to MASH development. So this shows the contributor to today's my talk, Kazufumi Matsushita, genetic knockout. He found that this is endoribonuclease important for inflammation. And he also involved ILC2 research. And Hideaki Iwasaki identified the degradation pathways. And Takuya Uehata generated this specific knockout. He identifies Regnase-1 is cleaved by MALT1. Hiroki Tanaka generated Regnase-1 knock-in mouse in which the IKK phosphorylation mutated the RNA. This he identified the IL-17 is also regulated Regnase-1. Yasuhara Nagahama regulated intestinal specific knockout mouse and also intestinal specific knockout. They identified the molecular mechanism of these, the phenotype of these knockout mouse. Takashi Sakaguchi generated liver specific knockout, and this mouse are highly resistant to MASH. Well, thank you for your attention.
Thank you very much. Yeah, I was super surprised with your summary slide with 13 results, which explaining how much mining you have done in this project. Well, fantastic. So thank you very much. And I'd like to close, but before closing, I would like to share one slide. Shizuo, could you stop sharing? Then I can share my slide. Okay, thank you very much. So let me share this slide. Okay, so if you want to make any question to Dr. Shizuo Akira, please go through the Twitter. And first, please search for account Global IMMUNOTALKS and find tweet that say ask question for Dr. Shizuo Akira here. And reply to that Twitter with your question. And please mention hashtag Global Immune. So that's all, and thank you for joining, and have a nice day.